Okay, thank you very much, Yossi, for this invitation. It's my first trip to Israel, and so far I've been having a great time. So this morning, or what's left of this morning, I'm going to be talking about the LINC complex, and by way of introduction, I just want to recap some of the major features of the nuclear envelope. So just bear in mind that the nuclear envelope contains an inner nuclear membrane and an outer nuclear membrane. The two are joined where they're spanned by nuclear pore complexes. Biochemically, these two membranes are really quite distinct. The inner nuclear membrane has its own unique array of membrane proteins. The outer nuclear membrane is continuous with and forms part of the ER and consequently has many of the features of the endoplasmic reticulum. Lining the nuclear face of the inner nuclear membrane, obviously we have the nuclear lamina, which we've heard a great deal about. And what I'm going to be focusing on a little later is this complex of proteins that spans both nuclear membranes, both the inner nuclear membrane, the outer nuclear membrane, and which we refer to as the LINC complex. Okay, so I don't have to talk about the nuclear lamins at all because Bob's told you about them. I don't need to say anything about all of these different diseases. The only reason I bring them up is to make the point that 13 or 14 years ago, in collaboration with Colin Stewart, we knocked out the lamin A gene in mice. And these mice develop a syndrome which bears a striking resemblance to human Emery-Dreifuss muscular dystrophy. So they have obviously dystrophic muscles, they have cardiomyopathy, they're small, and they invariably die within about 8 weeks of birth. Now, it's not so much the mice that I want to discuss this morning, it's the fibroblasts derived from these mice. So if we look at wild-type mouse embryo fibroblasts, and these are fibroblasts which are labeled with an antibody against the nuclear pore complex protein, NUP153, you can see that they have pretty much normal-shaped nuclei. Same for the hets. However, in these laminae knockout fibroblasts, you can see that these nuclei are very oddly shaped. Pore complexes are clustered, and generally speaking, they look peculiar. This, by the way, is another knock-in animal that I'm not going to talk about. It has dilated cardiomyopathy, but it has also seriously messed up nuclei. Now, in a way, the appearance of these nuclei are perhaps not so surprising. However, what is surprising, or what came as at least a big surprise to me, is that not only do these Lmna-null fibroblasts have mechanically unstable nuclei, it turns out that their cytoplasm also has altered mechanical properties. So this is taken from some paper from Denny Wirtz's lab, who were able to show using microreology that the cytoplasm of these Lmna-null cells is actually softer. It's less mechanically resilient. Jan Lammerding and his co-workers came to exactly the same conclusions several years before this, as did Jos Breuers. So basically the message here is that these Lmna-null fibroblasts, not only do they have mechanically compromised nuclei, they also have mechanically compromised cytoplasm. So what can be the basis of this? Well, it's been known for a long time that the nucleus and cytoplasm are mechanically coupled. And what I'm showing you down here is a figure from a paper from Don Ingber's lab at Harvard, where he was able to show this quite graphically. Basically, what they did was to poke cells with a microinjection needle and then move the needle. And what they were able to find was that the nuclei deformed in the direction of the movement of the nucleus. So the nucleus became stretched out. Furthermore, components within the nucleus also moved in the direction of the nucleus. So evidently, somehow or another, there are components of the cytoskeleton out here which are coupled to components within the nucleoplasm in here. So what's the basis of this mechanical this mechanical coupling? Well, logically, I think this has to be mediated, at least in part, by molecules associated with the cytoplasmic face of the nuclear envelope. So basically, proteins out here. So we know from Larry Gerace's work and Eric Shermer's in particular that there are at least 60 or so integral membrane proteins within the nuclear membranes, and several of them, and we now know, reside out here in the outer nuclear membrane. All of these outer nuclear membrane proteins feature a carboxy-terminal KASH domain, and I'll get back to exactly what that is in a few moments. All of these KASH domain or rather KASH domain containing proteins, at least in mammalian cells, include the nesprin family of spectrin repeat proteins, and we'll come on to those momentarily. Actually, we'll come on to them right now. So we know of at least 6 KASH containing proteins, or genes, or 6 genes encoding KASH domain proteins in mammalian cells. Now, I'd like to first focus your attention down here. What exactly is a KASH domain? It's a sequence of approximately 60 or so amino acids, contains a single transmembrane domain and a small luminal domain which pokes into the peri-nuclear space, so the space between the inner nuclear membrane, or the outer nuclear membrane, and the inner nuclear membrane. All of the KASH domains that we know about contain this proline cluster at the carboxy terminus. Okay. So what about all of these mammalian KASH domain proteins? We know of, as I say, we know of 6 genes. Nesprins 1 and 2, at least the largest isoforms, are absolutely enormous proteins. Nesprin 1, or the giant form of Nesprin 1, is about a megadalton in size. Nesprin 2 is 800 kilodaltons, so these aren't exactly the most convenient proteins on the planet to work with. They contain multiple spectrum repeats and exist in multiple splice isoforms, which essentially makes them an experimental horror show. The Nesprins 1 and 2 are both, or the giant forms are both actin-binding proteins. They contain an N-terminal actin-binding domain. Nesprin 2 also has a kinesin-binding domain, which is just down in this region of the molecule. Since we've been discussing various nuclear envelope associated diseases over the last few minutes, I just want to point out that that several disorders have been linked to mutations in the Nesprin 1 and Nesprin 2 genes. So Nesprin 1 mutations are linked to cerebellar ataxia, which may result from a neuronal migration defect. Both Nesprin 1 and Nesprin 2 mutations have been linked to forms of Emery-Dreifuss muscular dystrophy. Then we get onto this, this next group of proteins. Nesprin 3 is a plectin-binding protein, so it forms connections to the intermediate filament system. Other than that, we really don't know what it does at all. Nesprin 4, and this thing down here that we christened Dalek, I'll talk to you about in just a few moments. So what exactly are the mechanisms involved in the localization of KASH domain proteins to the inner nuclear membrane, or sorry, to the outer nuclear membrane? This is not a trivial question, since if you localize a membrane protein, or if you stick a membrane protein into the outer nuclear membrane, what prevents it from just drifting off into the peripheral ER? Well, the answer to this, or at least a possible answer, was suggested by observations on C. elegans, which came from, among other people, from Yossi Grunbaum's lab. So the focus here was on ANC1, a giant actin-binding protein, which is a prototype member of the KASH domain protein family. ANC1 is known to localize, or was known to localize to the outer nuclear membrane. Its localization was found to be dependent on a second membrane protein, UNC84. Now, UNC84 is a prototype member of another protein family, the SUN-domain family. Now, UNC84 is localized to the inner nuclear membrane, and its localization was dependent on the presence of the single C. elegans lamin. Okay. So what Yossi and others suggested was that, well, perhaps UNC84, this SUN-domain protein, functions as a transluminal tether for these outer nuclear membrane nesprins. Okay. Now, if this model was right, it has some quite staggering implications. The first, or the most important of which, is that these would form a pair of links in a molecular chain that would actually connect the lamina to the cytoskeleton. Okay. Okay, so what might be happening in mammalian cells? Well, we know that the human genome encodes multiple SUN-domain proteins. The most significant being SUN1 and SUN2. SUN1, needless to say, comes in a whole pile of different splice isoforms, the significance of which we still really don't understand. There are 3 additional SUN-domain proteins which are largely expressed in testis, although not completely expressed. But the SUN domains which are found in the majority of somatic cells are SUN1 and SUN2. However, there's even an exception to that, which I'll come back to in a moment. Okay. Well, instead of boring you with a lot of experimental data about this, we were able to confirm that both SUN1 and SUN2 do indeed function as transluminal tethers for outer nuclear membrane nesprins. We also know that SUN1 and SUN2 interact with components of the nuclear lamina. So exactly as Yossi and the C. elegans guys suggested, these SUN-nesprin or SUN-KASH pairs really do seem to function as links in a molecular chain that connects the cytoskeleton to components within the nuclear interior. So we christened these SUN-KASH pairs link complexes for linker of the nucleoskeleton and cytoskeleton. Now, recently, Thomas Schwarz and Ulrike Kutai actually sold the structure of the mammalian SUN2 SUN domain, and were able to show that it exists as a trimer. And trimerization is mediated by this extended coil domain, which forms a sort of triple helical stalk-like structure poking up from the inner nuclear membrane. Now, the KASH domain is able to interact with the SUN domain at the interface between two SUN domain monomers. So theoretically, a single SUN trimer would be able to interact with 3 KASH domains in theory. Whether that's the case, we don't actually know yet. I just also want to make this point, which was also taken from this Sosa et al. Paper, which is that the triple helical stalk, at the end of which we find this 200 amino acid SUN domain. This stalk, in principle, can extend about 45 to 50 nanometers into the peri-nuclear space. And this is sufficient length to span the distance from the inner nuclear membrane to the outer nuclear membrane. And many years ago, when we were originally characterizing these link complexes, we had knocked down SUN1 and SUN2 by RNAi and found, at least in HeLa cells, that when we did this, the outer nuclear membrane and inner nuclear membrane would expand. So the peri-nuclear space would become somewhat dilated. So at least in HeLa cells, it suggests that maybe these link complexes actually have a role in stitching the 2 nuclear membranes together and maintaining this even 40 or 50 nanometer gap. Whether that's true in all cell types is We don't know. So the nucleus and the cytoplasm are evidently mechanically coupled, but does this actually have anything to do with link complexes? So Jan Lammerding and collaborators, including us, addressed this point by sort of revisiting the experiments that were carried out by Don Ingber, which is basically to poke cells in the cytoplasm, move the needle and see if the nucleus deforms, exactly as you can sort of see here. And essentially what we found is as follows. In wild-type cells, if you do this, or in untreated cells, if you do this, what you find is you poke the cytoplasm, the nucleus deforms, it becomes longer and it becomes thinner. If we overexpress a dominant negative KASH domain protein, or if we knock down SUN1 and SUN2, or if we transfect these cells with a dominant negative SUN-domain protein, this is what happens. We completely, or not completely, but we significantly depress this coupling. So you poke the cytoplasm, you move the needle, and the nucleus and its contents remain largely undeformed. So evidently, these link complexes do indeed play a role in mechanically coupling the nucleus and the cytoskeleton. So what I'd like to do now is to focus on a couple of these other KASH domain proteins that have been identified over the last few years, and to describe to you, or at least try to describe to you, exactly what role they may be playing in actual cells and animals. Now the first one that we focused on is this protein nesprin 4, which we identified maybe 4 years ago. And we did this just by homology with the nesprin 2 KASH domain. We basically went searching in the computer for something that looks vaguely like this sequence, and indeed we came up with this sort of somewhat degenerate looking KASH domain, which we subsequently found was attached to this protein nesprin 4. Okay, so what can we tell you about nesprin 4? Well, it's a gratifyingly small protein for a start, which made us all extremely happy. When you transfect it into a human cell line, this is a human salivary gland cell line, but that's not really particularly relevant. When we put in this tagged protein into these cells, it localizes to the nuclear envelope, and this localization is absolutely dependent on both SUN1 and SUN2, or rather SUN1 or SUN2. You need one SUN protein or the other. And it's also absolutely dependent on the integrity of its KASH domain, this region here. Other than that, there's nothing particularly striking about this protein. It has a single spectrin repeat, a leucine-rich region, which probably forms a little miniature coil, but that's basically about it. However, one striking feature that we did notice, or one striking effect that we did notice, particularly upon expression in HeLa cells, was the following. When we introduced a GFP-tagged form of nesprin 4, what we found is that the nucleus and the centrosome moved apart, sometimes quite dramatically so. So we have this plotted out in this panel here. In control cells, what you find is that the centrosome usually resides within about 2 microns of the nuclear surface. The same if we just introduce a GFP KASH form. So this would be just GFP fused directly to the KASH domain. So the cytoplasmic business end of nesprin 4 would be missing. However, when we introduce full-length nesprin 4, we can see this fairly dramatic effect. The centrosome now, on average, moves about 12 or so microns from the nuclear surface. And it's not unusual to find the centrosome 40 to 50 microns away. So that basically means you have a nucleus at one end of the cell and the centrosome at the other end of the cell. So this is sort of quite a dramatic shift. Now, the other minor effect I want to point out here which you should remember for at least 10 minutes or so, is this. In many of these cells, what we find is that Nesprin 4 doesn't distribute evenly around the nuclear surface. It tends to concentrate on one pole. And this concentration of Nesprin 4 always occurs at the pole of the nucleus that's most distant to the centrosome. Okay. Now, going back to our centrosome movement, again, as you probably appreciate, there are a number of organelles which are associated with the centrosome also. So what we're doing here is looking at the distribution of the Golgi apparatus in cells expressing its GFP-tagged form of Nesprin 4. And what you see is the Golgi apparatus, just the same as the centrosome, moves away from its normal position at the nuclear periphery. And again, in many cases, you can find the nucleus at one end of the cell and the Golgi apparatus in the other, at the other. So the point I want to make here is that expression of a single outer nuclear membrane protein, a LINC complex protein, can actually cause large-scale changes in cellular organization, which, at least to me, was quite a startling effect. So what can we say about Nesprin 4? Nesprin 4, it turns out, is a kinesin binding protein, and we've been able to show this using a whole variety of methods. We know that the kinesin binding domain is in this sort of membrane proximal region. Now, kinesin 1 is a microtubule plus end motor protein. Will this kinesin binding function account for many of the effects we see associated with Nesprin 4 expression? So what I envisage as happening is as follows. We know that microtubules are anchored at the centrosome by their minus ends. Okay. If we now introduce Nesprin 4 into cells, it'll localize to the nuclear envelope and it will recruit kinesin to the nuclear envelope. So essentially Nesprin 4 is functioning as a cargo adapter for the nucleus. So kinesin at the nuclear envelope is going to engage with these centrosomal microtubules, and what's going to happen is this is going to cause the centrosome and the nucleus to move apart from each other, and anything that's associated with the centrosome is all, like the Golgi apparatus, is also going to move apart. So all of the experiments I've described to date described the fate of tagged Nesprin 4. So where is Nesprin 4 normally expressed? Well, I can't even read this, so I don't expect you to, but basically we find it in secretory epithelial tissues. So this is mammary tissue. Over here we have salivary gland, pancreas, and a few other things. But basically, Nesprin 4 is expressed almost exclusively in secretory epithelial tissues. Now, I'll say almost exclusively because in about 2 minutes I'm going to tell you that that's not actually correct. But just to make the point, this is an immunofluorescence analysis of some tissue sections. I've got to give credit to a former student of mine, Melissa Crisp, who basically sectioned a mouse from nose to tail to try and find where this thing was expressed. And lo and behold, she found it in pancreas, salivary tissue, mammary tissue, nothing in liver, and about 1 cell in 1,000 in kidney. Okay. So my sort of fantasy for a role of, for a function for Nesprin 4 was that maybe it has a role in epithelial morphogenesis. Maybe Nesprin 4 is required to move the nucleus or maintain the nucleus at the base of the cell. Now, the microtubule cytoskeleton in many epithelial cell types is non-centrosomal. What one finds is lateral microtubule bundles extending from the apical region of the cell down to the basal region. The plus ends of the microtubules are usually oriented towards the base of the cell, the minus ends up towards the apex. So my idea or fantasy was that, well, Nesprin-4, or rather kinesin, at the nuclear envelope would be able to engage with these microtubule bundles and keep the nucleus down at the base of the cell. So of course, to test this hypothesis, we went to the trouble of knocking out the Nesprin-4 gene in mice. So this just shows you the knockout construct. It essentially takes out the entire gene. This demonstrates quite clearly that, that Nesprin-4 expression is gone. You can see it in wild-type pancreas. It's completely absent from from the Nesprin-4 null pancreatic tissue. So my thought here, of course, was that the phenotype of these mice would have something to do with salivary glands, pancreas, mammary tissue. So maybe they'd have problems with their digestive tract or lactation. Of course, what we found was that they had absolutely no phenotype whatsoever that we could recognize. Story of my life. For about a year, these mice sat up in the animal house, just sort of eating away our budget, until I got a call from a friend of Yossi's here in Israel, Karen Abraham. Now, Karen is a human geneticist with an interest in hearing loss, and she informed me of something quite amazing, which is that she just mapped a form of progressive human hearing loss in a pair of Israeli families of Iraqi origin, and lo and behold, the mutation that she found was in our Nesprin-4 gene. So Henning Horn, who was a postdoc in Colin Stewart's lab, walked down to the animal house and basically did that above the cages, and there was no reaction whatsoever. So evidently, these mice are deaf. And to demonstrate that they're deaf, Karen carried out a whole series of auditory brainstem response measurements, and it is absolutely clear that these mice developed sort of profound hearing loss by about day 30. So we have deaf mice, and I've put this deaf mouse logo here for Ivan, because it's a punk rock band from Prague, which I thought you might appreciate. Okay, so what can we say about hearing in the mouse? Actually, I'm just going to pull up all of these images. So obviously, the business end of the auditory system is the cochlea, this coiled, fluid-filled tube within the mouse inner ear. And the important bit that we need to focus on, which is involved in sensing sound, is the organ of Corti. Now, the organ of Corti is rather complicated, but the important cells are these outer hair cells, which are present in 3 rows, which extend from the base of the cochlea all the way to the tip, and inner hair cells in a single row, which similarly extend from the base to the tip. And you can see a scanning EM down here showing the 3 rows of outer hair cells and this single row of inner hair cells. Okay. There are also multiple supporting cells within this structure also, which I'm not going to really talk about. So the question, of course, is Nesprin-4 expressed in the ear? Well, yes, it is. This protein here, prestin, is a marker for outer hair cells. I don't really want to say much about this other than that it's kind of membrane-associated, unconventional motor. But it's absolutely clear that Nesprin-4 is present in the nuclear envelopes of these outer hair cells, as well as in the inner hair cells. Okay, and this we're looking at 12 days after birth. What about in the knockout animals? Well, clearly Nesprin-4 is gone. from the outer hair cells. But what you might also notice here is many of the outer hair cells have also gone. Okay, so clearly there's something going awry here. So what might be the problem? So again, these are electron micrographs or scanning EMs provided to us by Karen Abraham, which clearly shows in the wild-type animals, looking at the mid-region of the cochlea, there's 3 nice rows of outer hair cells, and this is about a month after birth when we know that these animals are stone deaf. In the nesprin 4 null animals, there's no outer hair cells to be seen, although we can still see inner hair cells. At the apex, we can still see outer hair cells, but they're kind of ratty. Now, this loss of hair cells in this midsection versus this apical section is consistent with what we know of the features of this progressive form of hearing loss in both mice and humans. So basically, high frequency hearing goes first, then low frequency hearing. The bits of the cochlea involved in high frequency perception are in the basal regions, with low frequency perception out in this apical region. So the fact that we see this progression of hair cell loss from base to apex is absolutely consistent with the progression of hearing loss from high frequency to low frequency. Just want to make the point here that when we do tunnel staining of these nesprin 4 null cochleas, round about day P11, when we've still got a reasonable number of outer hair cells left, what we can see is numerous tunnel positive cells, suggesting that perhaps we're losing outer hair cells by a process of apoptosis. So this gets back to my fantasy of what nesprin 4 might actually be doing. Is it involved in nuclear positioning in hair cells? And the answer to that is, I'll give you in just a moment. Okay, so the point I want to make from this slide is that at birth, everything appears normal. There's a full complement of outer hair cells from both the base, or in this case the mid, to the apex. The other point I want to make here is that in the 2 weeks after birth, the auditory system matures, and the morphology of the outer hair cells changes. They start off sort of a bit cuboidal and then become sort of taller. Okay. All right. So this is what wild type outer hair cells look like. And we're labeling them with a bunch of different reagents. And you can see that the nucleus is sitting down here within the basal region of the cell. So what about in the knockout animals? Well, apart from the fact that the epithelium itself looks absolutely shot, just sort of focus on where the nuclei are. They're now up near the apex of the cell. So evidently there is some nuclear positioning defect associated with nesprin 4 loss. And you can see this happening over time. Again, I'm just going to load all of these. But these are z-axis reconstructions. At P8, you can see in both wild type and nesprin 4 null animals, the outer hair cells are sort of cuboidal looking, and the nuclei are more or less in the same place. As the cell elongates and the animals start to become sensitive to sound, what you find is that the nucleus in the mutant animals appears to migrate or fails to be retained down at the base of the cell. Okay. And this is just to make the point yet again, it's a segment of a single row of hair cells, and again in the wild type hair cells you can see the nucleus down at the base. In the mutants, it's migrated or floated up near the apex, and many of these cells develop a sort of bizarre hourglass shape. Now, as I hopefully made clear earlier on, the LINC complex comes in two parts. KASH domain protein on the cytoplasmic face of the nuclear envelope, and a SUN domain protein in the inner nuclear membrane, which functioning as a tether. So what one might predict is that if you can screw up hearing with with deletion of an outer nuclear membrane KASH domain protein, maybe you'll have the same effect if you delete its cognate SUN protein in the inner nuclear membrane. So hopefully you can see here that SUN1, it turns out, is the only SUN domain protein that we can detect in inner hair cells, in outer hair cells. Okay. So when we look at the distribution of, God, I can hardly see that, the distribution of nesprin 4 in SUN1 knockout animals, what you find is that nesprin 4 becomes completely delocalized from the nuclear envelopes of outer hair cells. What's also evident from this slide, which again is from a P25 animal, so it's is that its cochlear organization is completely shot. It's evidently lost a significant number of outer hair cells. Again, we can see the same nuclear positioning defect in SUN1 mutant animals. So the message here is that you can eliminate either end of this LINC complex, and you will have the same effect with the nucleus mislocalizing from the base to the apex of these outer hair cells. So I just want to summarize what we've found with nesprin 4. It's a kinesin binding protein of the nuclear envelope, expressed primarily in epithelial cells, including outer hair cells. It's associated with progressive hearing loss in humans and evidently in mice. Nesprin 4 deficient outer hair cells clearly have a positioning defect, as do SUN1 deficient hair cells. And evidently, a nesprin 4 SUN1 LINC complex is essential for outer hair cell maintenance. Now, the big question here, of course, is why mispositioning of the outer hair cell nucleus leads to cell death. That's something I can speculate on at length, but I won't speculate right now. Okay, so for the last few minutes, I want to talk about this last KASH domain protein that we christened Dalek, or sometimes Dalek 6, and I'll explain the terminology in just a moment. Again, we found this by homology, but by a much more circuitous route. It clearly contains a KASH domain, albeit a very degenerate-looking KASH domain. What can we say about this protein? Actually, not much. It has the carboxy-terminal KASH domain, a central coil domain, and a minor terminal EF hand-like sequence. If we stick it into HeLa cells, a tagged form into HeLa cells, it localizes the nuclear envelope in a SUN-dependent manner. Actually, these are P19 EC cells. But if we stick it into these cells, it localizes to the nuclear envelope in a KASH and SUN-dependent manner. So it's behaving exactly as a LINC complex, as a KASH domain protein should. One of the first things we noticed, however, when we introduced it into HeLa cells was that it behaves in exactly the opposite way to Nesprin 4. So you recall, in the case of Nesprin 4, it tends to concentrate at a pole of the nucleus that's most distant to the centrosome. This new protein, Dalek, tends to concentrate at the opposite pole closest to the centrosome. And this sort of suggested to us that, well, maybe it's not a plus-end motor protein binding protein, but rather a minus-end binding protein. And that turns out to be exactly right in both pull-down experiments and in transfection experiments. We can show that in this case, Dalek will pull down both dynein heavy chain and dynein intermediate chains, plus we can use it to recruit cytoplasmic dynein to the nuclear envelope. We were able to show that dynein binding involves this EF hand-like sequence, and that this coil-coil region is involved in self-association. So where is it expressed? Well, it turns out the only place we could basically find it was in testis. You recall in HeLa cells, it localizes very nicely to the nuclear periphery, pretty much like any other nuclear envelope protein that we've looked at. However, in testis, and in particular in spermatocytes, we find it, sure enough, in the nuclear envelope, but in these little patches. So what might it be doing? So again, this gives you an overview of the distribution of this protein. It's expressed almost exclusively in primary spermatocytes, so those cells which are undergoing meiosis. Now, what exactly is happening in these cells, and what do these patches correspond to? So you heard from this morning that during meiosis, chromosomes undergo homologous pairing. So what we're looking at here are synaptonemal complex proteins, SCP3 and SCP1. So SCP3 is a lateral element protein which sort of lines each homologous pair. SCP1 is a transverse element protein which sort of links the two together. I think it's instructive here to do a sort of comparison of the sort of microscopic techniques that we're using. So in this first panel, or first series of panels, we're looking at this this cell using conventional deconvolution, and you can see SCP3, this synaptonemal complex lateral element protein, forming these nice threads in the nucleus, basically where the two the homologous pairs are brought together. This is overlain with the SCP3 labeling. However, we've also used structured illumination, so a super resolution technique, which essentially improves the resolution by a factor of about 2. And in structured illumination, you can actually begin to distinguish each side, both threads of the lateral element protein SCP3 linked together by SCP1. So this actually provides you with a very nice way of determining whether your homologous pairs have come together or not, because you'll see 2 threads versus 1. Okay, so where is Dalek 6, or Dalek? Lo and behold, it localizes precisely to the telomeres, so to the tips of these synaptonemal complexes, which you can see using conventional deconvolution as well as in structured illumination. However, in structured illumination, you can actually see that it forms, or at least appears, as a little ring at the tip of each axial element. So when you have unpaired chromosomes, you see a single ring, and when they come together, you actually see a little double ring. Okay, whoops. Okay. So our prediction, of course, is that Dalek 6, or Dalek, would be this outer nuclear membrane KASH protein that's functioning as a link to telomeres in meiotic cells. So the mammalian version of what we heard about this morning, the KMS1 in yeast and Zyg12 in C. elegans. Now, what SUN protein might it be associating with? Well, it exactly co-localizes with SUN1 on the surface of these spermatocyte nuclear envelopes. SUN2 is completely absent from these cells. So the implication is that Dalek and SUN1 are associated at these telomere plaques or attachment plaques. Now, it's been known from work from Min Han from several years ago that SUN1 is actually required for telomere attachment to the nuclear envelope in mice. So if you make SUN1 knockout mice, their only obvious phenotype is that they're infertile, and this infertility is associated with loss of attachment of meiotic telomeres to the nuclear envelope. However, we also know that these mice are deaf as well, which these guys don't know, apparently. Okay. What exactly might be going on here? So you heard earlier on this morning about sort of bouquet formation and homolog pairing, so I really don't have to go into this at all. But what we feel is most likely to be happening is that this protein functions as an adapter as an adapter for cytoplasmic dynein. It's tethered by SUN1. This is an old slide, so this should actually read trimer rather than dimer. And SUN1 forms attachment sites for telomeres at the nuclear periphery. So you can see exactly what would be predicted to happen. Dynein would engage with centrosomally focused microtubules. This would then drag this whole complex, including telomeres, in towards the centrosome, forming a meiotic bouquet. So obviously, there are certain predictions one could make about what would happen if we knocked out Dalek in mice. So it would allow us to dissociate telomere attachment from telomere movement. The prediction is that in these knockout mice, the spermatocytes should retain telomeres at the nuclear envelope, since SUN1 shouldn't be affected. However, bouquet and homolog pairing should not, in fact, occur. Now, at this point, we decided to obviously knock this protein out in mice. I should have taken Yasusushi's advice and just stuck to yeast instead of torturing myself for the past 2 years with this. But basically, we were able to knock this protein out, and lo and behold, we got a phenotype which was more or less predictable. When we looked at testes from wild type versus Dalek null mice, you find that the mutant mouse testes are about a quarter the size of their wild type counterparts. In histological sections, you can see that while the wild types have normal-looking seminiferous tubules full of mature sperm, that's not the case for the knockouts. There's no evidence of sperm cells or spermatids whatsoever. In fact, many of these tubules look strangely small and largely acellular, with not much in the lumen. When we've carried out tunnel staining on these mice or on tissue sections, we find huge numbers of tunnel positive cells. However, this should not be taken to imply that there's apoptosis going on here, since there's a lot of other stuff that can be happening. Now, I'm running way out of time here, so I'm going to be very quick about this and make the point that what seems to be going wrong in these animals is that in the absence of this protein, Dalek, we can't form homologous pairs. So homolog pairing doesn't occur, and the spermatocytes arrest in early meiosis 1. Another prediction, of course, that you would make, assuming that this protein is behaving exactly as we think it should, is that in wild-type animals, you should see dynein co-localizing with these SUN1 telomere attachment sites, which indeed you do. And in the knockout animals, the dynein should be absent, which indeed it is. Okay. I'm not really going to go into this at all because it's getting towards lunchtime. But basically, what we were able to demonstrate here is we've knocked out this cytoplasmic half of this meiotic link complex. Essentially, what we find is that telomeres do indeed remain associated with these SUN1 plaques, although that's not 100%. I can chat to you about this afterwards. What is absolutely clear, though, is that homolog pairing never occurs. So somewhere in the transition from this conformation to this, these cells are arrested and ultimately die. So in summary, what we found is the functional homolog of C. elegans Zyg12 and POMB KMS1. It's a dynein binding protein expressed in mammalian meiotic cells, forms a meiotic link complex with SUN1. The knockout animals are gratifyingly sterile, and that's also true of females, although the gametogenesis in female mice is a big problem to look at. Okay, so I just want to acknowledge my many collaborators in this. My guys in Singapore, Graham Wright in the microscopy unit, and my former people at the University of Florida. I also want to acknowledge my long-time collaborator, Colin Stewart. We've been sort of working on lamins and nuclear envelopes and things since we were contemporaries at the EMBL more than 25 years ago now. Henning Horn has done a lot on both the Nesprin 4 and Dalek mice. Sergey originally made the Nesprin 4 knockout animals. Kyle and Dayin were also involved in both the Nesprin 4 and Dalek project. And of course, I have to acknowledge Karen Abraham and her group who really saved our bacon over the Nesprin 4 animals. So thank you very much.